Right, so here we're looking at the characteristics of cell membranes. All right, we're looking at a cell membrane right here, and we make up the cell membrane using structures called phospholipids. All right, so here we have a phospholipid, and here's a phospholipid. That phospholipid is made up of this head. We call this the phospholipid head, which is what we call hydrophilic. It means that it's water loving or polar. Okay, and then we have these tails, which are going to be two fatty acid tails that face away from the water. We're going to call those hydrophobic or water fearing. We also call them nonpolar. Okay, now this hydrophilic head is going to be made of what we call glycerol and a phosphate group. Okay, and this is because this is water loving, it's going to face toward the aqueous parts of the environment. So we're going to have an aqueous environment on the outside of the cell called the extracellular fluid, and on the inside of the cell called the intracellular fluid. So you're going to see these little hydrophilic heads that are going to be lining up next to that aqueous environment. Okay, again, the hydrophobic tails want to avoid water. They're water fearing or water hating, and they're nonpolar, so they're going to avoid it and they're going to line up on the inside away from the water. All right. Another term that you want to be familiar with is amphipathic nature. So when we say something is amphipathic, we're saying that it has both a polar region and a nonpolar region, or a hydrophobic or hydrophilic region and a hydrophobic region. All right, that's what amphipathic means. Now, phospholipids are definitely going to be amphipathic. You'll also see cholesterol here in the cell membrane. That's also considered amphipathic, as well as some proteins. All right, now this property is going to be really important because that ensures that we form this bilayer. All right, that bilayer, which is going to help with the integrity of the membrane as well as its function of letting substances in and out of the cell. Okay. We also describe it as being dynamic, having a dynamic nature. All right. Phospholipids are able to move back and forth. They're able to rotate. They're able to flip up and down, upside and down in between leaflets, which is rare, but it occurs when it has enzymes called flipases. All right, but when we say it's dynamic, it means that it's changing. If we say something is static, it means that it stays still, it doesn't change at all. All right, the cell membrane is not static. It's not like that. It is constantly changing, and you have these phospholipids that are constantly moving. Okay, now the function of this phospholipid bilayer, which is what we're looking at here, two layers, we call it a bilayer. The function of this is it provides what we say is a semi-permeable barrier. When we say semi-permeable, that means that some substances are able to go in and out, and other substances are restricted. As far as what's allowed to cross that membrane, we're going to see the smaller substances, the non-polar substances that are able to go right through this non-polar region of the membrane. All right, those are going to be things like gases, oxygen, carbon dioxide, and then we also have lipid-soluble molecules that can pass through freely. Okay, it will restrict large molecules. It'll restrict polar molecules, which mean that they have a charge. And you know, it those substances will be able to go through the cell, but they're going to require things like proteins. Okay, so they can't just go through the membrane here. The other function is that it's going to help maintain the structural fluidity of the membrane. All right, those fatty acids in the phospholipids will make it fluid and jello-like. The cholesterol makes it a little bit stiffer. All right, so that you'll see those inside the plasma membrane here, and that's going to stabilize this a little bit more. You know, you do want it to be fluid so it can kind of move and it can squeeze through small areas, but don't want it to be so fluid that it breaks apart. And that's where the cholesterol comes in, where it kind of gives it a little bit more structural stability, prevent that from becoming uh, excessively fluid, or even rigid. If there's too much cholesterol, those cells can become rigid.